Hello, my name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce our speaker, Dr. Mirko Corselli of Ph.D. Biosciences, for this webinar presentation titled Evaluating Expression Patterns of Multiple Inhibitory and Stimulatory Receptors on Natural Killer Cells. Dr. Mirko Corselli obtained a Ph.D. in Clinical and Experimental Hematology and Oncology from the University of Genova, Italy. He then transferred to the University of California, Los Angeles, to continue his research as a postdoctoral fellow. Most of his academic work has focused on the biology and therapeutic potential of human mesenchymal stromal cells. He is an R&D staff scientist at Ph.D. Biosciences in San Diego, where he is a flow cytometry subject matter expert and is also involved in developing innovative immunology and immuno-oncology applications. So without further ado, I hand the presentation over to Dr. Mirko Corselli.
Thank you for your presentation, and thank you everybody for attending our webinar today. I'm particularly excited to talk to you today about multicolor flow cytometry applications that we have developed for the study of heterogeneous natural killer cells. And just as a highlights of the webinars and what we will talk today, at the high level, we are going to talk about the resolution of natural killer cell heterogeneity and how we achieve these through multicolor flow cytometry analysis by simultaneously assessing stimulatory and inhibitory receptor expression on NK cells. And at the end of the talk, we also look into advanced high-dimensional data analysis tools that can enable the identification of difference from donor to donor when it comes to NK cell repertoire. As for the agenda, we'll start from a brief introduction on the biology of NK cells, their function and phenotype. We'll briefly touch upon flow cytometry, the basics and consideration for successfully designed multicolor panels. We'll then dig a little bit deeper on the data that we generated using two separate 12-color immunophenotyping panels. And we will wrap up by upgrading to a single 18-color NK cell immunophenotyping panel. Starting by describing the biology of NK cells are innate lymphocytes and represent the first line of defense against infected or transformed cells. They have the ability to directly eliminate infected or transformed cells through the secretion of cytotoxic molecules. They also have the ability to regulate the immune system through the secretion of immunoregulatory cytokines. The maturation and function of NK cells is tightly regulated by expression of surface and secreted protein. It can be broadly grouped into different categories, either a stimulatory receptor, inhibitory receptor, chemokine and cytokine receptors, as well as effector molecules. And for today's webinar, we'll focus our attention primarily on stimulatory and inhibitory receptors. And one of the most important features of NK cells is that they are highly heterogeneous and vary greatly from donors to donors. And that would be one of the main topics of this webinar, and how flow cytometry can help us revealing those differences. Looking a little bit more in detail about NK cell development, we'll use this cartoon from this review from Frontier in Immunology, where we know that over the years, over decades, a lot of study has been performed to identify the different step of maturation of NK cells from bone marrow, from a pro-NK phenotype, to mature NK cells in the circulation. And the fate of NK cells and the maturation state of NK cells can be mapped by assessing the expression of different receptors that define this subset. It's important that in flow cytometry we use surface markers primarily as a tag that allow us to call a cell based on their phenotype, but biologically all these markers, they exploit a very specific function that is necessary for the role that NK cells play in immunology. And for the for today's webinar, we're going to focus primarily on the mature NK cells. They're present in the peripheral blood. And in the peripheral blood, we find two major subsets of NK cells. The majority of NK cells in the blood, roughly 90% of the NK cells, are mature NK cells, which express a dim level of CD56, and they do express the marker CD16. A small percentage of NK cells in periphery, roughly 10%, are early NK cells, which are predominantly in the primary lymphoid organs, but they're still present at a small frequency in blood. And these cells are characterized by the expression of high level of CD56 and no expression of CD16. And if you look in the box, you can see that the expression of various receptors changes across these different subsets of NK cells. And just to bring attention on some of the markers that we will talk about in this webinar, if you look at the category of inhibitory receptor, you could see, for example, that the heterodimers of CD94 and NKG2A, it's more highly expressed on the early CD56 high NK cells, and it started decreasing over maturation. And an opposite phenomenon you will see in the expression of KIR receptor. These are killer immunoglobulin-like receptors that are not so expressed in the early CD56 high NK cells, and they're gradually overexpressed in mature cells. So you can really observe this pattern of expression as cells change through maturation. Speaking more about the cell function, we will focus on these stimulatory and inhibitory receptors because they're very critical to regulate the function of NK cells. And this other review from Long et al. From Clinical Cancer Research, it's a simplification here and a representation on how NK cells function. And you can see that if we are in a scenario where NK cells that express both inhibitory and stimulatory receptor interact with the target cell, in this case the target cell in the scenario A, they do express an MHC class 1. Now the inhibitory receptor, they bind to MHC class 1 molecules, and that allow to recognize self cells and normal cells. So in the presence of an inhibitory receptor binding to a normal MHC class 1, the NK cells, they're not activated, they don't trigger killing of the target cell. If an NK cells in scenario B encounter a cell that does not express MHC class 1, so there is no triggering of the inhibitory receptor. However, this is not sufficient to still elicit a full-on response of NK cells, because the target cell in this case is lacking activating ligands that will bind to a stimulatory receptor. So this tells us in the scenario C, for NK cells to fully be activated and trigger the cell-killing pathways, a target cells need to simultaneously lack MHC class 1 and also express activating ligands. And these are usually stress-induced or damage-induced protein like MIC-A, B, UBP1. They can now bind also to the stimulatory receptor, and that in conjunction to the lack of inhibitory signal trigger a full functional response and eventually killing of the target. If a target cell expresses both normal MHC class 1 and activating ligands, in that case we reach a dynamic equilibrium. So this is a simplified picture, but you can imagine that there is a great variability in the number of either MHC class 1 molecules that a cell can express and the activated ligands that a target cell can express. And that brings to the next level of complexity, that is about the heterogeneity and the diversity of receptors that an NK cell can express. We can classify inhibitory receptors with different families. We have the KIRs, and we have the heterodimers of CD94 NKG2A proteins, as we discussed before. We have different families of stimulatory receptors. You find on the right of this cartoon from the same review. We have different families. So we have the NCRs, the KIRs, the SLAM, the NKG2, and all the combination of all these receptors enables to basically have a large repertoire of NK cells that can respond to a variety of signals, either inhibitory or stimulatory. And that basically represents what is called the NK repertoire or haplotype. It contributes to the great heterogeneity and diversity of NK cells. A recent study estimated that just based on the mathematical combination and the possibility of the combinatory possibilities of expression of this receptor, each individual can have somewhere between 6,000 and 30,000 different subpopulations of NK cell receptor. And this poses some challenges on how can we study and fully understand the biology of NK cells and their diversity with such level of complexity. And that's why flow cytometry, it's a very well used tool for the analysis of NK cells because of its ability to measure simultaneously different parameters at the single cell level, as compared to a bulk analysis like could be a Western blot, where we can identify the expression or lack of thereof of a protein at the bulk level. A Western blot will not give us information on how many cells out of the total are expressed in the protein. So if we really want to quantify the frequency of cells expressing a specific phenotype, we really need to use single cell analysis like flow cytometry, as you see in the panel below. And flow cytometry has also become more and more relevant because of the evolution of flow cytometry, and now allows us to study a larger number of parameters simultaneously. As the hardware and the reagents have increased, the number of colors that we can look in a flow cytometer that expand our ability to simultaneously interrogate one cell at a time for multiple proteins. And that's the approach that we took in the study that we are discussing today. And we will start from, as I mentioned in the agenda, we'll start from describing a modular approach where we are designing 12 color panels. They are modular in their nature. And by that, we mean that we use a backbone with 6 markers. And these are 6 markers that are conventionally used to assess the presence and frequency of major NK cell subset based on the expression of marker like 56, CD16, as we discussed, CD27 expressing early NK cells, CD57 on more mature cells, and CD94. And this is going to be the core of our panel. It's a 6-marker core panel. To this panel, that we can add 6 additional markers. There can be a selection, for example, of stimulatory receptors, like the one illustrated in this table, or inhibitory receptor. And this approach has the advantage of providing flexibility. So we are maintaining the core of major NK cell identification markers. Then we can add 6 color for stimulatory markers, 6 color for inhibitory markers. And if we wanted, we could expand our analysis and keep adding different drop-ins, whether we want to look at chemokine receptor, cytokine receptor, or even intracellular molecule or effector molecules. So really this speaks about the flexibility and the value of having a modular multicolor flow cytometry panel. For the analysis of this panel, we started our panel design using a flow cytometer analyzer, a BD LSR Fortessa X20 system, equipped with 4 different lasers, a capability of 16 fluorescent parameters in total. And in this case, we adopted panel design best practices to choose our fluorochrome. And as is illustrated here, the check mark is the fluorochrome that we have utilized. On the right hand, it illustrates the spectra that we have available and how all the different laser and dyes excited by the different laser fill the space and eventually are utilized for designing this panel. And in this scenario like this, it's very important that, as you can see, there is a lot of overlap among the dyes. So the panel design, it's based on the ability to design a panel, taking in consideration the overlap between dyes, and make sure that we minimize the spillover, and which may affect panel resolution. And so with that consideration in mind, we design our 12-color panels. In this webinar, I will not go too much into the details of how the panel was designed step by step. We have additional webinars and tutorials that go deep into the panel design approach. I would just highlight that the major consideration that we're taking account for this panel design was first and foremost the biology. It's important when designing a panel that we are aware of the antigen density of our markers and the co-expression levels between different populations, so that we can allocate fluorochromes based on their spillover and their brightness accordingly. It's also important to have an optimized setup for the instrument. And while these rules overall work very well for a lot of model systems, in the context of NK cell biology, I would like just to make a general comment that antigen density and co-expression patterns in a model system like NK cells are not always that predictable. As we will see, some of the markers that we have selected greatly vary in their expression or co-expression patterns from donor to donor. So how can we account for that variability and minimize potential impact on the panel design? And for example, if we look at a marker like CD57 or CD127 that you can find in this table that we know they're highly variable across donor, we assigned what we call as a neutral fluorochromes, like FITC and B421, which are not greatly impacted by other fluorochrome, and they do not introduce spillover in other detectors. So even if we find variability across donors, that will not impact the rest of the panel. So that was just a little caveat that I want to emphasize for this story. So we have now our 6-color backbone panel, and then we have designed our drop-in stimulatory panel and inhibitory panel. So when we start looking at the data, we can start from the backbone panel, and that allows us, as I was mentioning, to resolve major NK cell subset. So we start from an exclusion of CD3 lymphocytes, T cells specifically, and within the lymphocyte gate scatter gate, we still look at NK cells, and the first gate that we can draw is the conventional CD56 on the y-axis and CD16 on the x-axis. And there you find those two subsets that we were discussing earlier, the early, more rare CD56 bright, CD16 negative cells. These are cells that are less cytotoxic. They're not very cytotoxic, but they express cytokines. And in the more mature cytotoxic cells with less cytokine production are the majority in this case, which are the CD56 dim, CD16 positive cells. Another way to classify the NK cell subset based on their maturation and function is to look now at CD56 on the y-axis and now CD94 on the x-axis. And you can really discriminate here on the top right a small population of CD94 high, CD56 high cells that correspond to those, again, early NK cells that, as we discussed, they express high level of CD94 and they will lose CD94 as they start differentiating. So we see an intermediate population right there in the middle where it's a CD56 high, and CD94 positive. And then we'll have the more mature population that will have pretty much lost CD94 or expressed very low level of CD94. From the backbone panel, we can still dig deeper into this subset. So if we now dig deeper into the CD56 high, CD16 negative, or CD56 dim, CD16 positive, we can look at other markers of NK cell maturation, and we confirm that, for example, in the early NK cells, there's no expression of CD57, and we have some expression of CD27. And we can observe a significant population of CD57 mature NK in the subset of CD56 dim, CD16 positive cells. So just from the analysis and combinatorial analysis of those six markers in our backbone, we can gather a lot of information about the distribution of different subsets of NK cells. But now we'll focus on the two subsets based on CD56 and CD16 expression, and we start looking deeper into stimulatory and inhibitory receptor based on our drop-in panels. And so on the top panel, you will see combinatorial analysis here of different combination of bivariate plots where we can look at stimulatory molecules either in the CD56 bright or CD56 dim population. And we can see different level of expression of this stimulatory panel in different subsets of T cells, notably CD160, in agreement with recent data in the literature. It's primarily expressed on the CD56 dim positive cells as compared to the bright cells. And also, as we showed before in the cartoon before, we have the NKG2C, the heterodimers with CD94, which is more expressed on the early NK cells as we expected. So this also gives us a good representation of the good resolution of the markers that we achieve in a 12-color panel and allow us to look at different patterns in different subsets. If we look now at the other tube where we have the 12-color inhibitory panel, we can also appreciate here again resolution and specificity. For example, if we look at the KIRs, the inhibitory receptor CD158A and CD158B, in agreement with the biology and with what we know about the maturation of NK cells, these receptors are not expressed in the early NK cells, but they're clearly expressed in the CD56 dim population. And on the other hand, NKG2A, it's more expressed in the CD56 bright as compared to the CD56 dim population. And really tells us again that we have not only good resolution, but also biological validity, and we are observing the patterns that we expect. Another level of information that we gather from this data is about the heterogeneity of NK cells. It's something that we discussed in introduction, but we are looking now at real data, and we can look that, for example, just if we look at CD158A and B, we have different combinations. So we have NK cells that express only CD158A or only 158B, or a subset of NK cells that express both or neither. So again, that speaks about the heterogeneity of NK cells and the different combination of receptors that we can find on a single cell, at the single cell level. And that brings us naturally to the next part of the webinar, where we now are going to step up to an 18-color panel. And why do we want to do that? It's because instead of separating a stimulatory and inhibitory panel, we can now put all these markers together into a single cell tube. And beside, of course, the workflow efficiency and the fact that we're running just one tube, these greatly expand our biological understanding of NK cell heterogeneity, because now we can look at all these different combinations of both inhibitory and stimulatory markers at the same time. However, the the challenge of going from 12 colors to 18 colors is because it's linked to panel design and the difficulty of somehow designing a panel with more markers. So if we go back to our configuration that we use for our 12-color panel, we can see that we have used 4 lasers and dyes excited by 4 lasers, and how we already have a very crowded spectral space where we feed all our dyes. Now, if we want to step up to our 18-color panel, we're going to take advantage of a Fortessa X20 equipped now with an additional laser, which is the UV laser. And there are multiple advantages on why doing so. Of course, the first advantage is that we have now more parameters that we can look at. We can look at now 18 colors, so we can look at more parameters. But most importantly, having this additional spectral space that we can utilize out of the UV laser allow us to spread out our dyes across a larger spectral space and spread out the 18 colors so that we can minimize the spillover. And that allows us to have an easier panel design because now we have more choices, we have more opportunities and more dyes that we can utilize, and increases our quality of data when we can spread out the dyes and minimize the spillover. So for example, you can see that we can use combination of these dyes. We can reduce the crowded space of the yellow-green and allocate some of the dyes now to the UV laser line. In our configuration, we can look at 4. In our specific configuration for this webinar, we could look at 4 UV dyes simultaneously. And it really gives us the opportunity to, again, distribute the dyes, minimize spillover, facilitate panel design, and get better results. It's also very important that a configuration like this is not only relevant to 18 parameter panels, it also allows you to have a much easier and much better resolution when you go to 12 or 14 colors, because now we can spread and minimize the spillover even more. It also introduces to a new family of dyes, which are the brilliant UV dyes. They have their own unique features and capabilities. Just give you an example of one of the dyes of the family, which is the brilliant UV 395. And this is what we call a spread analysis or an n-by-n analysis, where on the x-axis, you can look at CD4 staying with BUV 395. And what we do here, we are looking on the y-axis and all the other channels and all the other detectors in our instrument. And what you can appreciate that this dye provides a very good resolution of CD4 and introduces basically no spread into any other channel. So this is a very clean dye that we can now utilize. This is very relevant, for example, when we put markers at the top of our hierarchy on markers that are very highly expressed, and basically there's a high level of co-expression, high level of expression, and we can minimize the impact on all the other markers that are co-expressed. So this was just to give you an introduction on one of the brilliant UV dyes. This next slide speaks about the concept we were talking about, having more flexibility now in your panel design. So panel design, we take in consideration the biology, the expression level, the fluorochrome brightness, but there's also a limiting factor sometimes is the reagent availability. And when we have a lot of redundancy on the formats and on the dyes that we have available for each specificity, that might impact our ease of panel design. So just by introducing now the UV laser and the brilliant UV dyes, you can see that for these specificities, we have increased the number of available reagents that we can now choose multiple formats and more formats, and that allows us to basically have a greater flexibility in designing our panel. So with all those considerations in mind, we went ahead and designed our 18-color NK panel using the 5 laser configuration. And you can see that we have basically now each dot represents a laser line. We'll be able to spread across 5 lasers with taking consideration the antigen density, the co-expression levels, and we have optimized our panel to analyze simultaneously all these markers. But the first thing that we wanted to do, of course, was to make sure that biologically we are gathering the same information as we were when we were using the 12-color panels. And on the top, you see the 12-color panels and the definition of some of the subsets that we discussed, the major subset based on CD56 and CD94, the inhibitory receptors and stimulatory receptors. And you can see that biologically this is the same donor. We're getting equivalent results, an equivalent appreciation of the different subsets when we have now multiplexed all the panels into one, and we have changed the fluorochrome, but we still maintain the ability to detect those subsets. It's also to understand that when you increase the complexity and the number of markers, you expect resolution to be somehow impacted. That's expected. But again, the rule of panel design help you minimizing those impacts and making sure that even if a marker like CD56, in this case, you might appreciate is a little bit bright, is a little bit dimmer on the second panel, we still can detect those markers clearly and call those cells based on the phenotype. So this is great. Now we have 18 parameters. We have all our inhibitory and stimulatory receptor in one panel. But again, this poses an additional challenge. Another challenge that comes with expanding our panel from a 12-color to an 18-color is the complexity of the analysis of the data that we are going to generate. The more parameters we have, exponentially increase the complexity and the combinatorial possibilities of the markers the expression of the markers that we're looking at. And so the conventional flow cytometry analysis that it's based on basically a biased approach of manually gating on population based on a priori knowledge has a lot of drawbacks, like the subjectivity, the variability, reproducibility, but also inevitably introduces a bias. So as we move forward now with the evolution of flow cytometry in a higher and higher dimensional space, we also need to take advantage of computational flow cytometry analysis. And this is based on algorithms that can assess all markers simultaneously in a completely unbiased fashion. So we're going to give some example of these tools, and these tools can help both on the visualization of the data, but also on the identification of population that we have not been otherwise been able to identify. And to really show the full potential of a computational flow cytometry analysis, we have run that 18-color panel now on 5 individual donors. And in this case, we are getting on total CD56 NK cells. And we are using t-SNE analysis as a dimensionality reduction tool that basically take all the information and all the expression of the markers and reduce the visualization and the allocation of this marker into a bidimensional space. And it creates what now we refer as clusters or islands. So what we can do, and this is analysis that is performed on FlowJo, when we look at all donors, the first plot on the left, this is coming from a concatenated file. It means that we have merged the files from 5 individual runs and 5 individual donors. Now we have merged them into a single file. We can create a t-SNE plot, and now we can tease out each donor and see their clustering and their distribution of cells. And what is very obvious is that if you're now looking at the individual donors outside of the concatenated file, is that donor 1 through 5, you can see unique patterns and unique clustering patterns. They're very specific to each donor. Now, what t-SNE does, it's a visualization tool. So it's telling us that there's something in each individual donor and some markers that are differentially expressed and drive this different clustering. Now we need to start digging deeper and realize what are the markers that are driving this difference. So one approach that we can take is, for example, using a heat map, where now we can maintain the clustering and the islands that we see there, and we can interrogate one marker at a time and see through a heat map for each donor where the marker is expressed. And this gives us basically a spatial distribution of each marker, and it can also code it in a heat map fashion, so based on the level of expression. So for example, we can appreciate different CD57. For example, donor 3 has a predominant clusters that are CD57 positive and higher expression as compared to the rest of the donors. Donor 5, if you look, for example, at CD94, you can see that right at the right of the plot of donor 5, there's a very unique population there. Seems to be unique to that donor. It's very highly expressed in CD94, and similar different patterns if you look at CD158A. However, this is more of a special distribution. It's not going really at the single cell level. Another way that we can now look at all the markers in all the donors is conventionally looking at histograms. And so this is a very quick way to interrogate the level of expression and the patterns of each individual marker across donor. So we can again confirm in this case that CD57 is very variable across donors. Some donors express low levels, some donors express very high level. CD94 seems to really stand out in this case, where donor 5 has a high level of frequency of CD94 positive cells as compared to other donors. So this gives us some hints on what possibly drives the different clustering of different donors. But ultimately, the limitation of using single analysis like histogram is that we are not really, in this case, understanding the relation between different markers. We're looking at one marker at a time, but we don't know if these markers or some of these markers are co-expressed or not co-expressed. And so what's the combinatorial expression of these markers? So to really eventually close the circle here and dig deeper into this kind of analysis, we took advantage of another algorithm and another analysis tool, which is called SPICE, which is an acronym for Simplified Presentation of Incredibly Complex Evaluation. And what does this really tool allows you is to analyze large multicolor flow cytometry data and organize them again in a graphical visual way, very easy, but easily display in comparison of distribution patterns. And most important about the combinatorial analysis of receptors. So we're looking in all these receptors. You can remember for the introduction with the cartoons where you can express either an inhibitor or a stimulatory receptor or both of them, and the level of complexity when you look at all the different receptors. So just to give you an example of what we can do now from the same data set from the 18 color panel across 5 different donors, we create now the SPI and ARC charts. And let me just spend a couple of minutes just to go through the data representation if you're not familiar with this tool. But what we're looking here, we've selected 6 markers out of all the 18 that we have, just to simplify the analysis for this webinar. And we selected 3 inhibitory and 3 stimulatory receptors. We actually have CD94, which is an interesting one because it heterodimers with either NKG2A, CD159A as an inhibitor, but could also bind to heterodimers with dimerized with 159C as a stimulatory. In this case, we're going to look at the combination with the inhibitory CD159A. And the way we read this graph, if we look, for example, donor 1, each pie slice represents a subset of NK cell, and each subset of NK cell is defined by the combinatorial expression of these 6 markers. And that combinatorial expression is represented by the arcs outside of the cell. So we can see that there are arcs that they overlap with each other, and that would be co-expression. So we have population that express only one marker. Like if you look at the green arc, you could see at 9 o'clock of the first donor. At 9 o'clock, you see the blue pi slice. Those are a subset of NK that express only CD158, sorry, CD160 as a green arc. But if you look next to that arc around 8 o'clock in the chart, you can see that those NK cells now express both the purple and green arc. So that means cells express CD160, but also CD158B, and so forth. And you can really visually assess here how different and unique each donor is, thanks to the ability to analyze all these markers simultaneously. We can also quantify these, and you can really now see all the different combinations that we have at the bottom of different markers and see the different distribution across donors. We go back to the donor number 5, if you remember, had that very distinct cluster that was CD94 positive. So this donor specifically has that big slice, pie slice in orange, that's driven by the co-expression of CD94A and CD94 and CD159A, as also represented in the chart. You can see that big blue bar standing out the way. So again, this give us a lot of biological information on the distribution of markers, but most importantly also on the unique NK cell repertoire across different donors. So just to summarize what we've discussed in this webinar, we have showcased how multicolor flow cytometry can really allow you to resolve major NK cell subset and different functional and developmental stages of NK cell maturation. We have taken different approaches to study the expression of stimulatory and inhibitory receptor. We have utilized first a modular approach with a 6-color backbone and then 6-color drop-ins to study either a stimulatory or inhibitory receptor. But we also showed how taking advantage of different configuration of instrument and the use of the UV laser line with the brilliant UV dyes that allowed us to further expand our panel, facilitate the panel design up to 18 color, but most importantly, really enrich the level of biological information that we gathered. And that was also facilitated by the use of computational analysis, in this case performed on FlowJo or through the SPICE software. And all of this culminated eventually in the ability to easily identify donor-specific differences, NK cell diversity, using the tools that we have described. So, and with that, I would like to conclude by acknowledging the team that has been involved in this study. I'm a member of the R&D Immunology application team, and we are part of the scientific affair, medical affair, and application group. The team is led by Suraj Saxena, and for this study, the study was overseen and supervised by Stephanie Whitman, and the experiment was executed by 2 senior scientists on the team, Chad Sisomantong and Nian Kara. And I would like to thank you for your attention, and I'll be glad to take any questions you might have. Thank you so much.
Thank you, Dr. Corselli. So our first question is, how do we know if the panel tested provides the necessary resolution?
Yes, thank you for the question. So there are a number of ways that we can address this question. And I think that it all comes down to the controls that we need to include in our panel, panel design. One of the best way to analyze the panel and diagnose the panel, if you want to assess the resolution, you might want to have your single stain control for each one of your specificity. And then if you just do an histogram overlay or line up, compare your single control stain and the resolution of that stain, meaning the separation between the negative and the positive signal. And then you compare the analysis of the same specificity in the panel. Inevitably, when we look at 18 color panels, we will see an increase in the spread of the negative population that will reduce the resolution of the negative and the positive. And that's absolutely normal. We cannot avoid that phenomenon. What we want to ensure is that even if we're introducing spread as a normal function of spillover, we're still resolving the positive population. If by any chance we see that as compared to a single control, when we have now the fully stained panel, our negative population overlays with the positive or reduce the resolution, that would be an indication that maybe that choice was not the best, and identify then who introduced the spread and change the panel accordingly.
Okay. Next question would be controls. What are the controls needed when designing an 18-color panel?
That's another great question. It's a good segue to the previous point. I mentioned the single stain control. That's always a good control to have in your panel, not only for the compensation, which I always recommend to use the cells as possible, same with the specific antibody, same lot of antibody, especially for tandem dyes. That it's good. The single stain control, as I just mentioned, it also can be used, so as a dual function beside compensation, can also be used to assess the impact on resolution when we compare the single control to the fully stained panel. And a related control that is always good for multicolor flow cytometry panel, beside the single stain control, will be the fluorescence -1 control, or FMO control. And this control, again, is critical as a dual function as well. So basically, this control, it's a tube where, if we are looking at 18-color panel, will contain 17 markers, 17 fluorochromes, and will exclude just one at a time. And we will do that for all the specificities. How do we use the FMO control? One way, the most common way to use the FMO control is to properly set our gate. Because if we use an unstained control, we will not take an account of the spread that is introduced by all the other markers in the tube or in the panel. If we use an FMO control, now we can really look, for example, at FMO for 50, we set our gate, we looked at, there will be spread introduced, and we can set the gate now taking account that spread. The other important role of the fluorescence -1 control goes back to the diagnose of the panel and see if we have necessary resolution. Let's say that we have identified that in one of our channels, the spread that was introduced was too high, and now we cannot resolve our positive. How do we know who introduced that spread? How do we know who was the culprit? And that's where the FMOs can be used. We can just look at all our FMO controls and tubes, and at some point in one tube, we'll see that the spread of the negative population will go back to normal, and our resolution will be restored. And if you look at the tube that was FMO BB76, that tells us that BD76, in the choice of assignment to do the antigen that we use, was the one introducing the spread. So basically, I would recommend when setting up these experiments to have your single control and your FMOs. Now, the FMOs is not something that you may want to do all the time. It's something more on when you optimize your panel, you want to make sure that the panel works, you want to diagnose the panel, and then you don't necessarily need to do it for all the donors. Having said that, though, since we're speaking here about this model system where we have high variability of markers, the FMO controls for one donor might not be appropriate for a different donor if there is a marker that greatly varies among donors. So again, it all goes back to knowing your biology, and if you know that there's 2 or 3 markers in your panel that they are highly variable, if you want to really be accurate, include the FMOs just for those 3 or couple of markers that are unpredictable, and that will put you in the best conditions to have accurate and confident data.
So related to that is, so you have your 18, you've designed your 18 color panel. How do you really ensure and know that the instrument that you're using has the optimal setup for that panel?
Yes, that's another great question. It goes back to those 3 pillars of panel design, knowing your biology, knowing your dyes, but also making sure your instrument is properly set up. There are different ways that can be done. And what does the optimal setup mean? It means that the instrument is, the PNT voltages on the instrument are optimized such that we can get the maximum resolution possible for each of our specificity, or for each of our fluorochromes. And the way we can do that, it's one approach that we can use is called voltage titration, where we basically can take, for example, CD4, stain in all the different colors, and we acquire the same tube with the same specificities and the same fluorochrome at increasing voltages. And what you will see when you analyze your staining index, which will be a function of resolution, and it will tell you how well separated your signal is, you will see a typical curve, and then your staining index will plateau. And that means that at that point, you're not gaining any more resolution, likely because your negative populations start increasing together with your positive. So basically, you want to set up at that point, mark down what the PNT voltage is, where you reach the plateau, and that tells you that your instrument is providing you the best resolution for that specific marker. Now, one consideration would be that this is the optimal resolution. It's fluorochrome and antigen independent. It doesn't matter if it's CD4 or CD8 or CD19. We're just measuring the performance of the instrument. Having said that, if, for example, we are now using in our panel a specificity that has an antigen density higher than CD4, there's the risk that our voltages might be too high for the specificity and our signal will go off scale. So what I would recommend is, even though your instrument is set up optimally based on a voltage saturation performed on CD4, whenever you optimize a panel or you diagnose a panel, before you even run all your controls and your compensation, quickly look at your sample and make sure that all your signals are off scale. It will be, if you start compensating and you're 3/4 of the process it would be really hard to change the voltage here to rerun all the controls. So quick check on the voltages and on the signal and make sure everything's on scale, and you can utilize the best optimized settings of your instrument.
All right, great, thank you. So one question actually related concerning equipment is, can this panel be prepared and used on a fax lyric system?
That's another great question. Just to clarify, the fax lyric is a 3-laser, 12 parameter, up to 12 parameter instrument. So the exact panels that we showcase here, the 12 color or the 18 color panels cannot be utilized on lytics just because of the different configuration and the lack of a yellow-green or a UV laser. Having said that, we have actually designed lytic-specific panels for these markers, for these inhibitory and stimulatory panels, using a similar approach of backbone plus drop-ins. The short answer is exactly the same. Panels cannot be used on lytics because of the different configuration, but they can be designed for lytic. And once you are comfortable with the panel design process, at the point the rules and the consideration to be taken are configuration agnostic. Whether you have 2 lasers, 3 lasers, or 5 lasers, you will use the same process to design your panel. And lytic is it's a good example of a 3-laser instrument with great resolution. And as a matter of fact, these panels can be designed on a FACS lytic. To that point, in the FACS lytic, we've also been able to expand the concept of the drop-in that was based on what I showed you in the introduction, where we complemented the backbone panel not only with inhibitory and stimulatory receptors, but we're also able to expand other tubes with cytokine receptors and chemokine receptors. And again, speaks about the flexibility of flow cytometry. It's agnostic to the instrument as long as you are comfortable with the panel design process and you follow the rules. And of course, the limitation goes back to the biology. The advantage of moving up to anything color is how deep in the biology you want to go. How do you want to correlate your biological information in a multi-parameter fashion? But that doesn't take away from having smaller panel or smaller tubes that you can do based on the configuration that you have available.
Okay. I believe that is the end of the question and answer time that we have for today. I would like to thank Dr. Corselli for taking the time to give his presentation and answer these questions. This webinar will be posted on the Current Protocols website at currentprotocols.com for viewing later on. And once again, just thank you, Dr. Corselli.
Thank you, everybody.
Goodbye.